Hey biology students, we're starting a new unit now. The title of Unit 4 is Biodiversity and Ecology. This week we're doing lecture 17 on biodiversity of life. In this first lecture video, we're going to discuss the history of life and the first cells. So we're going to start by taking a little field trip back in time 4.6 billion years ago. This is the scientific recognized age of the Earth. However, if we did go back in time 4.6 billion years ago, the Earth would look significantly different than it does today. So although water was available, there was absolutely no life forms in the early days of Earth history, primarily because there weren't the available biological molecules just yet. In fact, there was no oxygen in the early atmosphere of Earth, although there were lots of volcanic eruptions, and based on fossil evidence, scientists believe that the early atmosphere of Earth was primarily carbon dioxide and nitrogen gas and methane gas. So it's hard for us to comprehend these huge timelines in geological time, but about 1 billion years goes by before biological molecules form, including DNA, RNA, and protein, and organize in membrane-bound little bubble cells that would become the very first cells on Earth. So it's recognized that the first cells on Earth were bacteria. And specifically, we classify these cells as anaerobic. That word means surviving without oxygen. And this was important, of course, because the atmosphere of early Earth did not have oxygen. So timeline here, we are about 1 billion years after the origin of Earth, about 3.8 billion years ago, the first cells, the first prokaryotes, which were bacteria. If you recall from our unit on cellular respiration, we talked about the amount of ATP that's produced by cells when they break down food molecules by cellular respiration. And we talked about briefly in that unit that some cells utilize anaerobic respiration or fermentation pathways to make the ATP they need for cell survival. If you recall, anaerobic pathways do not produce very much ATP. Now that's okay if you're just a single-celled bacteria living on the surface of a warm bubble in the or in the ocean. But in order for multicellular complex life forms to evolve on this planet, which is what we see today, a significant change in the way organisms use their food molecules would have to take place. And what happened was the biological evolutionary process began to take place in that very, very first group of prokaryotes. So even among prokaryotes, genetic variation caused by random mutation occurs. So a random mutation allowed for a new group of prokaryotes to evolve on Earth. Now, these prokaryotes are still around today, okay? So the descendants of these prokaryotes are still alive today, and they're in a group of bacteria we call the cyanobacteria. And what's really special about this group is that they can photosynthesize. This, in fact, is the origin of all photosynthetic organisms on the planet. And so they evolve, and as a result of photosynthesis, which we have also studied in detail in this course, oxygen is a very important byproduct of the reaction of photosynthesis. So this process, now beginning to develop in huge populations of bacteria, allowed for oxygen to begin accumulating in the atmosphere. And we recognize that timeline at about 2.5 billion years ago. Now, this allows for aerobic bacteria to evolve by random mutation and selection. Remember, aerobic means surviving with oxygen or in the presence of oxygen. And cell respiration in the presence of oxygen is way more energy efficient than anaerobic respiration or fermentation. Remember, aerobic respiration, these organisms produce between 36 to 38 ATP per glucose, compared to anaerobic respiration, around 2 ATP per glucose. It's equivalent to if somebody said, would you like to work for $2 an hour, or would you like to work for $38 an hour? Sort of a no-brainer, right? You make way more cash for the cell. Remember, ATP is analogous to currency that the cell can use to cash in to do cell work. So this is a huge selective advantage to be able to survive and use the oxygen. And remember where it uses that oxygen, uses it in the electron transport chain step of aerobic cellular respiration. So this is a huge advantage and would explain how these aerobic bacteria began to develop. And this would also give rise to multicellular complex life forms that haven't evolved yet on my timeline here. But let's just appreciate this moment in history where oxygen is accumulating, cell respiration, the ability to use oxygen to make significantly more ATP occurs in these very simple celled organisms, but this is a trait that will be passed on to all of us and will allow for complex life forms like ourselves to evolve. Now, eventually, eukaryotic cells evolve. About 1.5 billion years ago in the fossil record, we see evidence of the first eukaryotic cells. And it's these cells, of course, these eukaryotes, that will give rise to all of the complex eukaryotic life forms that you are aware of and think about when you think about life, and you think about Earth, and you think about plants and animals and fungi, and you take a nature walk outside, and that's what you see. And that's a result of these very important early events that occurred in the history of life. So today, even though the first cells were bacteria, the descendants of those cells are still around today. Now, they branched into different groups that eventually would give rise to multicellular life forms, but the prokaryotes are certainly still alive and well, and very in huge numbers everywhere in the environment and on your skin and inside your body. And so we're going to pause a minute in the history of life, and we're going to talk about the bacteria in a little bit more depth. So as far as the classification of life on Earth, we classify life into 3 domains. And this is based off of cell structure. So there are 3 domains. And so let's describe the first domain. The first domain is the domain bacteria, okay? And it's classified as prokaryotic or prokaryotes. So what are some features of the bacteria? Well, they're all unicellular, and that's a word that means single-celled. And these are microorganisms that you need a microscope to see. They're incredibly diverse, so they do represent groups of anaerobic cells that are the descendants of those very first bacteria that lived 3.8 billion years ago. Many of them also respire aerobically, and many of them also perform photosynthesis and are descendants of that first group of photosynthetic bacteria billions of years ago. Recall they have a very simple cell structure. At the beginning of the semester, when we covered cells, we briefly looked at the cell structures in prokaryotes, but mostly we focused on eukaryotes back in those lectures. So at this point, we're going to talk a little bit more in depth about prokaryotic cell structures. So first of all, remember prokarya means before nucleus. So these cells evolved before the nucleus develops within cells. And so a key feature of prokaryotes is that they lack a nucleus, although they do have DNA. But the DNA of bacteria is a single circular loop. Okay, so they have a single loop of chromosomal DNA, still chemically identical to human or eukaryotic DNA in that it's still double-stranded and it still has the sequence or the alphabet of A, T, G, and C, but the organization, the chromosomal arrangement, we consider to be circular. It's a circular loop. How would we describe the eukaryotic chromosome structure? Well, recall when we looked at chromosomes in our mitosis and meiosis lectures, they looked sort of like sausages, right? And then they would replicate and look X-shaped. So that's what we call linear shape. So eukaryotes have a linear shape to their chromosomes, where prokaryotes have a circular or loop shape to their chromosomes. No membrane-bound organelles. Okay, so within a bacterial cell, you're not going to find a Golgi, you're not going to find an endoplasmic reticulum, you're not going to find lysosomes, no internal membranes at all. However, you are going to find ribosomes. So ribosomes are composed of RNA, a special type of RNA called ribosomal RNA, and protein. So we do find specific bacterial-sized ribosomes. Most bacteria have a cell wall. That's actually how we classify them based on their cell wall structure. Reproduction of prokaryotes is asexual. In other words, there's not a there's not a sperm or an egg that combine to form a new offspring, which is what we have in sexual reproduction. In these early cells, these very simple cells, they simply divided by cloning themselves in a process we call binary fission. This is a very fast process where the cells can double as fast as every 20 minutes. They can make an identical copy of themselves. It's similar to mitosis. So when we studied mitosis, recall that involves a single cell that clones itself or makes an identical copy that's genetically the same as the original cell. So it's similar to that, although it's definitely faster. There's also a high mutation rate in many bacteria, so we do get occasional genetic changes that can drive the evolution of bacteria. So they're not always 100% identical. What we're seeing here in this video, we're seeing a time lapse of a bacteria called Streptococcus pneumoniae, which is the cause of bacterial pneumonia. And you can see that it starts as a single cell, and begins to divide and divide. So one cell becomes 2, and 2 becomes 4, and 4 becomes 8. That's the type of division we call exponential. So they're exponentially increasing. This gives you an idea of how quickly bacteria can populate themselves, especially if it's a pathogen. So pathogen is a word we use for disease-causing organism. And most of you, if you know of bacteria, bacteria sort of have a bad reputation among us humans, where we think of them in terms of causing disease or causing illness. And those, of course, are the pathogenic bacteria, but they really only represent about 1% of all bacteria on the planet. So lots of bacteria are important for various reasons. They help in the decomposition of organic material. They produce some of the oxygen from the atmosphere by photosynthesis. They produce certain antibiotics that we use in medicine. And they also are a large component of your body system. So there are bacteria that live in your mouth, in your intestines, on your skin, in your ears. And those are actually part of a large collection of organisms we call your microbiome. And more in microbiology, we're realizing the importance of having sort of good bacteria, or what we call probiotic bacteria, beneficial bacteria that are part of your natural body systems that help in many ways, including digesting food and producing vitamins for you, and also keeping out pathogenic bacteria. On this slide, we're seeing the structure of a prokaryotic cell. Just wanted you to take a closer look at that. And once again, let's emphasize some of those key features of prokaryotes. So they do have DNA, okay? So they do have DNA, they have a chromosome, and a lot of times we use this word nucleoid as a region in the cell where that DNA is located. But it's not membraned enclosed. There's no nuclear membrane around it, okay? So no nucleus, but is oftentimes referred to as nucleoid. Also, let's note here that we have a circular loop. That doesn't really seem apparent in this particular picture. So let's draw it like that. However, in the lectures on biotechnology, I talked to you about plasmids. Plasmids are also circular loops, and that does look very clearly circular here in this structure in green. This is very common in bacteria for them to have extra chromosomal DNA. So besides their main chromosome with the main instructions for building their proteins, they have these additional loops of DNA we call plasmids. They're just smaller, and they encode fewer genes. So ribosomes are also pictured here, these little black balls. Okay, those would be ribosomes. We see that it does have a plasma membrane or a cell membrane, a cell wall, and many additional features not shared by all bacteria, but some bacteria have flagella, this tail-like structure here, or they have capsules or slime layers or little sticky hairs along their surface that they use for attachment. On the right of the screen, we're looking at the 3 general shapes for bacterial cells. So bacteria are really classified a lot of times by what we call their morphology, which describes their cell shape. So a lot of times they look, we call these ones in green. Of course, they've just been stained that color. Okay, that's not their natural color. We call this shape bacillus or rod shaped. Sort of look like Tic Tacs under the microscope. Other bacteria have sort of a ball shape or a spherical shape. Like you see, these look like sort of a cluster of purple grapes. This shape is called cocci. Okay, so it's a sphere. And the third shape is a spiral shape. This here on the bottom is showing you the bacteria that causes the sexually transmitted infection known as syphilis is caused by a bacteria with a spiral shape. So that's the domain bacteria. Now, prior to 1970s, scientists, thought there were pretty much just bacteria and then there was everything else. Until one day, a scientist discovered an entire new domain of life. And it was a headline on the front page of every newspaper in the world in 1977. Carl Woese, the American scientist, discovers a third domain of life. He discovers it accidentally, as a lot of great science is just accidental. Taking samples from deep sea hydrothermal vents at the bottom of the ocean, his research team identified these single-celled microorganisms that were being collected from the bottom of the ocean in these deep thermal vents, very hot, very anaerobic, thought to not be able to hold any life forms. But lo and behold, these small microorganisms were found. Now, he calls them the Archaea, and that comes from the word archaic, which in the English language means ancient. And at the time, he proposed that the domain Archaea was perhaps even more ancient than bacteria, which was the other group of prokaryotes known at this time. However, further experimentation has helped us to understand that the Archaea are actually descendants of bacteria. They are not older than bacteria, but the name has stuck. The Archaea, or the ancient microorganisms. The second domain of life, okay, also prokaryotes. So let's talk about their features. Basically, so very similar to bacteria in that they are unicellular, they are prokaryotic in their cell structure, and they reproduce by that binary fission, asexual reproduction method. They are known to persist and survive in extreme environments and have adaptations that allow for that survival. We call the ability to survive in extreme environments extremophiles. So they are known as extremophiles. So surviving in extremely hot or cold or salty conditions. Although that's not their only locations. So genetic analysis gives us a lot of information as scientists about the evolutionary relationships among organisms. And a very common way to analyze the DNA of organisms is actually to look at their ribosomal RNA. And it's called a 16S ribosomal RNA analysis. So it's a type of genetic analysis. And so when scientists performed the 16S ribosomal RNA analysis on the domain Archaea, they figured out that it was more closely related actually to eukaryotes than prokaryotes, even though it does have a prokaryotic cell structure. So we think they are descendants of the first cells being members of the domain bacteria. They're actually more ancient than the Archaea, and then next the Archaea, and then following the Archaea, we would have the third domain of life, which would be the domain eukarya. At this point, I would like to put all of the domains together in one place for us. Do you remember who drew this little sketch that's showing up on the screen? We've seen it before. That's right, we saw it last week, didn't we? In our lectures on evolution. So this is a little sketch, a little family tree of life, sketched by Charles Darwin. And remember, he writes, I think, at the top, and he sketches this little tree, and he proposes that all life on the planet has a common origin, a single-celled origin that then branched into multi-groups, all with their unique characteristics, and then that begins to evolve into more complex life forms. So at the time, Charles Darwin didn't have a lot of evidence to support his claim that life has a common origin and has been evolving for billions of years. Today, there is widespread evidence of this, and it's a large component of the theory of evolution. So one of the things we do in evolutionary biology is we put together something called phylogenetic trees. So Charles Darwin sketched here in his notebook the very first phylogenetic tree. We define it here as a map of evolutionary relationships among living organisms, including their ancestors, and it's based on fossil evidence, anatomical evidence, and DNA or genetic evidence. Okay, so let's take a look at how we would create a phylogenetic tree today to represent the three domains of life. To do that, I'm going to have to erase Charles Darwin's little tree there. Replace it with modern tree. Okay, so it is considered a tree. Now you can draw a phylogenetic tree either from the bottom of your paper up, okay, just like a tree, like if you would draw a tree, the understanding is that a tree has roots that sort of anchor it, and then that tree grows in an upwards fashion where the leaves at the top of the tree are younger than the roots, right? So you can certainly draw a phylogenetic tree in that way. In fact, that's how I'm going to do it right here. But you can also draw it from left to right, where you're moving in order of time from left to right, or you can move from bottom to top. I'm going to do a bottom to top one. So usually when we draw a phylogenetic tree, we include something called the root, okay? So we would put a little root here of the tree here. And this root would represent the first cells, the origin of cells on this planet, which is understood to be 3.8 billion years ago. We use that symbol BYA to represent billion years ago. Okay, and so those first cells branched into the three domains. So going off to the left from this branch, this would represent the domain bacteria. This includes the first cells, who we also know were bacteria, and all bacteria that evolved since then. Okay, so still around today. But then they branched into domain Archaea. So remember, based on the genetic analysis, and the word phylogenetic has that word genetic in it as sort of the major way we classify life by comparing DNA sequences and noting where we see similarities and differences. So although domain Archaea are also prokaryotes, they're going to be closer to this branch here that's going to lead us towards domain Eukarya. Okay, so those are the three domains of life and how they organize in a phylogenetic tree. So something that distinguishes bacteria and archaea from eukarya. Okay, so let's actually make a notation here that prokaryotes would be bacteria and archaea. Okay. However, the cells in the domain eukarya have a very different cell structure. And the origin of some of their unique cell structures occurred by a very important event. So a lot of times on these phylogenetic trees, you see these little notches on the tree, and it represents a very important evolutionary event. So the event that occurs here in the timeline of life is an event called endosymbiosis. Which we're going to describe in the next video.